Uh, my previous lesson was focused on the general framework of the database that could be included in a possible exposome analysis. Now I would like to focus on a more specific point that is also more related with my current activity, essentially the integration of structural and functional information for exposome analysis. In particular, I would like to say that now I am working on development on a molecular sensor for pollutants based on design new macromolecules such as nucleic acid and proteins with high specificity for contaminants. Data integration at molecular level is we have talking about this in the previous lesson, then I can skip because essentially basic data information, basic data about pollutants, characteristic, genomic and transcriptomic and so on. I guess that this framework is quite clear after these 4 days of course. Okay, this is an integration of transcriptome and proteome workflow, experimental workflow, that we have used in a European project for drug discovery. Then you can see the DNA, the genomes. In the middle is the black box, how to correlate the transcriptome to proteome. Now there are myrnomes and other information, proteome and animal model testing, and outside you can see the aim is in this case is the same workflow is modified in terms to analyze environmental toxical factors, mainly chemical factors. Okay, then the previous probably is less complete, or I forgot to delete the slide. You can see also the microRNA there. One of the big problems is to link proteome, myrnome, because you must simultaneously analyze the 3 different levels. This is the basic integration of functional data for exposome or for drug discovery. The difference between exposome and drug discovery is quite subtle, because one is for therapy and the other is for toxicant. A drug is a toxicant that it is used for therapy, but it's toxic in any case. Okay, you can see the integration that we have done. Original genome centers, this is the general pipeline. This all has been derived from this paper that I guess is until now very interesting and maintain its proper specificity and value. Okay. This gives an information about the quality of data that are generated from this kind of functional genomics. A single cell single cell molecule, the complexity of the study is relatively small. At cell biology level, the complexity is moderate because the intervening some factors such as cell lines and culture protocols, clinical phenotypes, height, dosimetry, ethnicity, and all the things that Professor Pasardani has shown in the example about Alzheimer. Drug response very high, and drug response very high could be considered as an aspect of exposome. Instead, the drug you put pollutant response. And this is susceptibility is very high because, as you can see here, the quality duration of exposure of environmental factor and the last not well defined economical and ethical aspect depend by health policies. In environmental toxicogenomics area, we are talking in the previous lesson, and it's I am recalling some key point because in one side there is the chemical informatics pipeline, and on the other is the genetic pipeline that in both must converge to the prediction of adverse effect. Okay, you can see the local data warehouse that we have used for the European project. The philosophy can be modified according to your project, but in general these are the basic protein for basic information that include array express that are functional genomics, HUGO human proteomics is electrophores to the gel to the pager, and the PDB that are structural, and the scope. In the previous lesson I was talking about structural elements. Scope is a database that contains the classification of basic structural proteins. Okay, this is one of the interesting things is to find the association between protein and Myrna, and to predict in which way the combination of protein and Myrna impact our metabolic pathway. You can see here the microarray and antibody array that is similar by fluorescence approach, and each one require a different data mining system. Is different from microRNA. The analysis is different in considering microarray, microRNA and protein. You can see here the role of exposition. We are going to the genes, exposure, stress, oxidative stress, methylation, stone coded, all the processes that contribute to reach or to determine the risk assessment for a disease. Okay, now the idea, the second step, having a pRNA is a PubChem toxicant. In this case, has been used for RNA structure. We are using PDB structure. We are using for docking, and we are using classification. We obtain a report. You can see in this case we have used, in this approach we have used, for instance, we have used these air pollutants. I here reported the PubChem code for simplicity because the names are very long. You can see now, you can see here the feature that we have used for a classification. In which way we have extracted these features. One of these features, the black features, are obtained by docking algorithm, are parameter estimated by docking. The other are data, the red are information extracted from databases such as PubChem or Zinc or other. It's important to understand that it's possible to embed in this analysis until 400 different features, because a chemical structure, and it will be the topic of my last lesson, is characterized by 400 descriptors. 400 descriptors are 400 features that can be used for classification. Normally are used by chemical physics features, molecular weight, rotable bond, hydrophobicity, what you want, and plus some information obtained by molecular analysis. Then in this data set, in this set are included some experimental data such as X log P and the number of rotable bonds that are experimental also determined, but a large amount donor and acceptor are known from experiments, and the other are the black essentially are in silico data. Okay, you can see, for instance, here the results of application of simple k-means cluster, that is the basic cluster methods, and in this case you can find the centroid elements. The centroid is considered that the vector, that the object that minimizes the distance from the weight vector. In this case we have used all these features, and in the first class we can find the N-nitrosmethylamine as a pivotal element of the cluster. In the class 2 is the P-phenylendiamine, and the C3 is the bis-chloromethyl ether. I would like to warn you that that this docking was carried out with not enough performing systems. By using other more performing systems, such as with using flexible ligands or other options, the result could be a little bit different or more different. Now we're talking about non-coding RNA. Non-coding RNA is a class of regulatory elements. They are ribosomal RNA nucleic acid, and they have different options. They participate in the ribosomal action. They transfer RNA that is related to the ribosome and the translation process. Small nuclear RNA are regulatory elements that stay in the nucleus. Small nuclear RNA that control the transcription generally, and that control point variation or chemical variation of information on macromolecule. MicroRNA are very well known. The last one, the short interfering RNA, is also well known. This class of compound is important because there are many kits for short interference experiments. One of the problems at this level is there are two problems or two constraints on the method. One is the specificity of the probe. The other are the off-target effect. What are the off-target effects? The off-target effects are all the factors that influence the hybridization action, the hybridization kinetic, due, for instance, to the ionic strength, molecular crowding in the cells, or other factor that is pH, or what you want. Delivery systems, because a short interfering RNA require a carrier system such as liposome or other to be transferred into the cell. Okay, you can see, for instance, this analysis was carried out on a sample of air pollutants that was obtained by a survey made near Nagpur, and you can see the experimental analysis has given information about the chemicals in PM10 pollutants. I would like to underline that many studies about PM particulate pollution normally talking about PM10, PM2.5. But inside this term and inside this element is a blend of elements in which there are pollens, chemicals, ions, and other substances, bacteria, virus, and what you want. Removing the biotic stressor, virus, pollen. The other are ions and chemical or organic chemical compounds, volatile, that are volatile and then can also persistent. This problem